Doug came to UCSD after completing his residency at Johns Hopkins University. He came in 1987 as a dementia fellow, working with Bob Katzman. And it was clear from the start that Doug was somebody we wanted to keep in San Diego for sure, and were able to. So in 1990, he joined the faculty in the Department of Neurosciences and became a VA attending physician, rose through the ranks, and is currently, since 2000, has been professor in residence in the Department of Neurosciences. He also had a stint from about 2000 to 2012 as chief of the neurology service at the VA hospital. And most importantly to all of us, from 2007 to 2016, Doug was director of the Shiley-Marcos Alzheimer's Disease Research Center. And then pulled back and decided just to be head of the clinical core, and he's been head of the clinical core of the Alzheimer's Disease Center since then. He's also extremely accomplished, has more than 400 peer-reviewed publications in all the important journals in the field, and he is currently himself co-editor-in-chief of a journal, Alzheimer's Research and Therapy. He's been involved in Alzheimer's research over the past 30 years. He's been involved in the development of clinical criteria for related disorders. He was a key mover in the original development of clinical criteria for dementia with Lewy bodies, and also in frontotemporal dementia. And his biggest claim to fame, though, is in the area of development of biomarkers for Alzheimer's disease. So he was one of the early leaders in the development of CSF biomarkers for Alzheimer's disease. And in particular, he was one of the first investigators back in 1993 to help show that a decrease in CSF A-beta 42 was a marker of Alzheimer's disease. And fortunately, we have him here as our own in-house expert on biomarkers. So today he's going to talk about CSF and blood biomarkers for Alzheimer's disease and update. So Doug, take it away.
Well, thank you very much, David. So I'm going to give everybody a tour of where biomarkers have been heading in the last year or two. These are my disclosures, and I'm going to cover a number of things, all relatively superficially. I'll start with a tour of biomarkers in general, talk about the classic CSF biomarkers, tell you what's been going on in blood biomarkers more recently, talk about why we need to diversify CSF biomarkers, and then talk about some of the recent exciting data on protein seeds. So to start off with, I think biomarkers are critically important, and they could have had an impact on history, as this slide would show you. So if we think of major causes of dementia in the elderly, most of what we know comes from pathology, and we know that Alzheimer's disease is the leading cause, but there are other degenerative and vascular disorders that contribute, and we would like to have a situation where we don't have to rely on autopsy to be able to make a specific diagnosis. We also know that it's possible to stage Alzheimer's pathology, in particular using the hallmark lesions of amyloid and neurofibrillary pathology. And these are slides from one of the original studies of the BRACs, showing how amyloid progressively spreads over a number of areas in the brain, some of which are part of the default network, and neurofibrillary pathology spreads from transentorhinal through limbic and isocortical regions, and neurofibrillary pathology correlates with cognition. I would also emphasize that although plaques and tangles are very important, especially as we deal with older individuals with memory and other cognitive disorders, they're not the complete story, and co-pathology is frequently encountered. And trying to attribute cognitive change to plaques, tangles, and some of these co-pathologies can be a challenge. We've resorted predominantly to clinical pathological studies, but we're hoping as biomarkers evolve and expand to be able to do these sorts of studies in living patients. So to summarize the co-pathologies, a number of vascular changes are important in the brain with aging, including microinfarcts, strokes, amyloid angiopathy, and so forth. Alpha-synuclein pathology is present in at least 20% of people who have Alzheimer's disease in their brain, and obviously synuclein pathology can be its own entity. And in the last few years, there's been increasing interest in TDP43 inclusions in different brain regions, and sometimes TDP43 can be its own pathology, laying waste to the hippocampus in hippocampal sclerosis. No slide, no talk about biomarkers would be complete without some reference to a biological definition of Alzheimer's, the work that Cliff Jack and others have pioneered. And so this was the latest framework called ATN, proposed in 2018, where the notion was that there's Alzheimer's pathological changes, which would include amyloid and tau pathology. The coincidence of both would define Alzheimer's disease. But a number of concomitant non-Alzheimer pathological changes were also important and could interact, and one could have non-Alzheimer pathological changes on their own. And so synuclein, TDP43, and vascular changes fit in there. And the green over here is cognition. And many of us believe that cognition is critically important, especially if you're talking about dementia. And the notion is that cognition would form a spectrum ranging from normal through subtle cognitive decline, mild cognitive impairment, and dementia, and that the pathology would build up during all of these different stages, and there will be some sort of relationship between the pathology and cognition. So part of the idea of the ATN framework is to try to start by defining Alzheimer's disease specifically using biomarkers. And the biomarkers selected would be a biomarker of amyloid, or A, a biomarker of tau, or T, to define the characteristic lesions, and then some or other biomarker representing the effects of amyloid and tau on the brain, which would be N for neurodegeneration. And N is an enormous category. So whereas one could start to hone down on amyloid and tau, one can imagine all sorts of different things living under this big tent of N. So ATN would decide that if we're going to approach clinical staging, we would use terms such as normal, preclinical AD, MCI due to AD, and dementia due to AD, whereas without biomarkers, we would use terms such as normal or MCI or dementia, and it would be harder for us to fit the appendage of AD to that terminology. So in terms of the kinds of biomarkers that can be used to map the ATN framework, for amyloid plaques, we can use amyloid PET or CSF A-beta 42. For tau fibrillary pathology, tau PET is really the best established biomarker. For neurodegeneration, we can look at anatomy, MRI, to show us atrophy and pathways, FDG PET to show patterns of regional glucose use, and then biochemistry, we can look at CSF tau and p-tau, which I think fit better into a neurodegeneration framework than into a tau fibrillary pathology framework, as I'll discuss later, and then other markers such as neurofilament light and a lot of other things that could be encompassed under, etc. So how do we decide that something qualifies as a biomarker and gets checked off? There's a regulatory interest in doing this, and I'd encourage people who are interested to look at this BEST framework that was proposed by the FDA in 2018, because it defines the context of use of a host of different biomarkers. And it will define, for example, that a biomarker can be used for diagnostic purposes, for monitoring in relation in particular to therapeutic interventions, predictive markers, prognostic, and so forth. And there are very specific categories that can be quite helpful. If we're going to apply this to Alzheimer's disease, we might think of screening or diagnosis being important questions. Diagnosis in particular, if we're looking at unusual presentations, early diagnosis or differential diagnoses, or trying to figure out if someone has Alzheimer's when there may be a situation of mixed diagnoses. In clinical trials, biomarkers have been transformative. They're currently being used to confirm that somebody actually has Alzheimer's if they enter a trial. They can be used to identify people who are asymptomatic,
next thing,
but have pathology for prevention studies.
Catch up, and we've got 2 more calls today together.
Robert, you're on.
Yes, 2 more calls.
They can be used to track or predict progression.
Yes, that's good.
Bye.
They can be used to evaluate target engagement and downstream effects, and can be used as outcome measures. So that's a quick introduction. Let's talk about the classic CSF biomarkers for Alzheimer's. As David mentioned, A-beta and tau were some of the earliest of the CSF biomarkers that have been discovered. And there are different forms of A-beta that can be measured, likewise for tau. But we're hoping to be able to broaden the map of biomarkers to, in particular, reflect processes that contribute to AD and neurodegeneration. So one might think of biomarkers reflecting inflammation originating from microglial activation, such as TREM2, astrocyte markers such as YKL40 or GFAP, markers of vascular and blood-brain barrier dysregulation, synuclein biomarkers, markers of general neuronal injury, neurofilament light I will talk about, and markers of synaptic dysfunction, which I will also cover. If we think about amyloid and tau, over the years, we've been able to develop quantitative and specific assays. CSF A-beta is produced by the cleavage of the parent protein APP, and this results in a number of peptides ending at amino acids 37, 38, 40, and 42 of the A-beta sequence. CSF tau is much more messy. These are data from a study published from Kai Blenau's group about 2 years ago, in which they showed that there are a host of different fragments. These are names of different antibodies that can recognize these fragments that can be found in the CSF. And these are all cleavage products of tau. There's no full-length tau that could be identified in CSF. And the C-terminal end of tau is critical for aggregation and for formation of tangles is not represented in CSF. So what we might be measuring may reflect some indirect processes relevant to tau pathology. So having said that, this is a summation from Forum. Bob Olsen in Sweden has curated a meta-analysis that started in 2016 and is updated from time to time on Forum. And this shows a host of different biomarkers measured across different laboratories and studies with more or less effect sizes and difference, which represent the difference between Alzheimer's and controls. The ones for which there are the most robust data shown in green here, a reduction in CSF A-beta 42 and the ratio of 42/40, an increase in total tau, which represents a number of those fragments I showed you, an increase in phosphotau, there are different phosphosites that can be measured, and an increase in neurofilament light. Some of the other markers do show differences, but they're less robust. So in terms of CSF A-beta 42, a large number of studies have now shown that the reduction in CSF A-beta 42 or in the 42/40 ratio is reflected by an increase in fibrillar amyloid, which can be assessed through amyloid PET. So the two are inverse markers of one another. A number of studies have suggested that CSF A-beta 42 starts to drop before amyloid PET crosses the threshold for positivity. And if one thinks of using CSF A-beta 42 in tau, this is one of very many studies. And it illustrates that if one looked at people who started out in this study with CDR0, people who had the highest risk to develop MCI or dementia over many years were those who had abnormal CSF A-beta 42 and tau and subtle memory decline, represented by this red line. The blue line is people who had abnormal A-beta 42 and tau together without some subtle cognitive decline. And then the other markers represent other intermediate risk states. So one of the advances in the last few years has been towards fully automated CSF biomarker assays. There are several companies that have developed these. At present, Elixis and Lumipulse assays are the most widely used, and they both use a chemiluminescent readout. They can deliver results very quickly. This is an example of the Elixis system, which produces results in about 20 minutes. But the major advantage is that there is low variability both within samples and across laboratories. So the Elixis platform has been used to measure CSF A-beta 42, tau, and phospho-tau in ADNI. And these were data actually initially published in both ADNI and the Swedish Biofinder study. And one can see that diagnostic classification, looking at some of the individual biomarkers and particular ratios of biomarkers to be able to classify amyloid PET regardless of the diagnostic status of the patient was extremely high. And these were some of the cutoffs that were proposed using the Elixis system in these studies. One could also look at ADNI data to decide whether using biomarkers in MCI had predictive value for which patients might progress to Alzheimer's disease. And in this study, it was shown, again, this is based on Elixis data, that people who had low amyloid had an intermediate but increased risk. People who had low amyloid and high tau had a markedly high risk, as shown over here. Having said that, there still is quite a lot of variability, wide error bars, and there may be room to improve and refine these models of prognosis, which I will come back to later. Let's switch and talk a little about neurofilament light. Neurofilament light is a structural protein found in axons, and it's a highly abundant protein, and its levels increase in CSF non-specifically in a variety of conditions that cause axonal degeneration or damage. So this is a composite from a series of studies showing that some of the highest levels can be found in frontotemporal dementia and some other similar types of disorders, corticobasal syndrome. Alzheimer's is a little increased compared to controls, but less dramatically than some of these other disorders. The panel on the right shows what would happen to A-beta 42 in these disorders, where Alzheimer's disease is associated with the decline, but most of these other disorders are not. Behavioral variant FTD can sometimes have Alzheimer's co-pathology or sometimes be misdiagnosed, and that might be a reason for the partial overlap. This is a meta-analysis of CSF neurofilament light data across a number of disorders. So one comment is that CSF neurofilament light increases with aging. And so whatever kinds of diagnostic cutoffs or thresholds one is going to determine should be normalized for age, and age should be taken into consideration. And again, this showed similar sorts of findings to the previous slide I had shown, where some conditions, in particular frontotemporal dementia, in this case HIV with major cognitive disorder, multisystem atrophy, corticobasal syndrome, are associated with marked increases. Alzheimer's disease is represented in orange. There is an increase relative to controls, not quite as dramatic as some of these other disorders. So that's kind of where I would let things sit with the classic CSF biomarkers. And I'm going to talk about some of the recent advances in blood biomarkers. Why has it taken so long to develop blood biomarkers for Alzheimer's and neurodegenerative disorders? One of the fundamental problems is that we'd like to be able to say that what we're measuring reflects the brain. And so the big question is, can proteins that originate in the brain be detected in the blood despite dilution? The blood volume is many-fold higher than the CSF volume, and clearance. There are lots of mechanisms in blood to get rid of things. So for example, plasma levels of A-beta are about 1/60th of those in the CSF, and they already are relatively low in CSF. There also is a short half-life of plasma A-beta, and it can be cleared by the liver and the kidney. Tau levels in plasma are extremely low. Neurofilament light, while the levels in CSF are reasonably high, neurofilament light is made both in central and peripheral neurons, and deciding that changes in plasma originated from the brain may be complicated as a result. So one of the things that's changed the landscape of blood biomarkers is the advent of ultrasensitive assay methods. And so to give you an example, these were first applied to neurofilament light. The concentration of neurofilament light in plasma is of the order of a low number of picograms per mil. So in Alzheimer's disease, there is a difference in neurofilament light compared to controls, although there is some overlap. In ALS, there's a massive increase. Guillain-Barré syndrome, which is a peripheral demyelinating neuropathy, there's a marked increase. And in some of the atypical Parkinsonian disorders, there is an increase, but if you look at the scale, it's been shifted. So we're talking about an increase with the units being measured in somewhere between about 25 and 50 picograms per mil. So nevertheless, the advent of these highly sensitive assays has made a big difference.
Doug, in GBS, did they distinguish between the axonal forms and the demyelinating? Sounds like it looks like a bimodal distribution.
I don't think they did, and I believe those are the demyelinating forms. So in Alzheimer's disease, in the ADNI study, it was possible to look at both plasma and CSF. And one can see that there is a relationship between plasma and CSF neurofilament light, although the correlation is intermediate. The R was 0.59, so it's not a perfect prediction. If one looks at how well plasma neurofilament light worked in ADNI, in people who were MCI and amyloid positive, determined by PET or CSF, there's a group increase, and it's significant versus controls, but there's clearly a lot of overlap. And if one looks at Alzheimer's dementia, again, there's a group increase versus amyloid negative controls, but again, quite a degree of overlap. So plasma neurofilament light would not serve as a diagnostic test for Alzheimer's disease, but perhaps it could be used for other purposes. And so because ADNI collected longitudinal plasma samples, it was possible to look at how neurofilament light levels changed over time. And if one looks in this first panel, remembering that it does increase with aging, so you're going to see a little bit of a trend. There's some degree of an increase in MCI, but a definite increase by between 4 to 6 picograms per mil per year in Alzheimer's disease. And this was more marked if people were amyloid positive. Not a huge effect of adding ATN status to this. And one can look at a number of other kinds of relationships. The major point was that these changes would start to begin somewhere in MCI, but are much more apparent in Alzheimer's disease. So perhaps it could be used to monitor interventions at the stage of Alzheimer's dementia. Let's turn to tau. So initial efforts to measure tau were based on some of the total tau assays. And initial success was found in people who had undergone traumatic brain injury with quite marked elevation after acute and severe TBI. This is a study looking at professional ice hockey players where they consented to have a preseason blood draw for plasma tau. And this was 1 hour post-concussion or 12 hours post-concussion. So there's a little bit of a bump that then tends to flatten. So it suggests that brain injury could lead to some small increase in plasma tau. Plasma tau has been inconsistently altered in MCI and Alzheimer's disease. This was a study that I was involved in with Dominic Walsh, who raised a panel of several different tau antibodies and suggested that a particular combination of tau antibodies could detect a form of just regular tau that might be increased in MCI and AD. There were two different cohorts studied, one with rather young patients at University College London and Mass General, and then from our ADRC. And there are significant differences, but there's quite a bit of overlap, and the levels are still extremely low. This is 2 picograms per mil versus about 4 picograms per mil. So a lot of data has now gone into trying to quantify the tau fragments that I'd shown you. Much of the work has come out of Randy Bateman's group with Nick Barthelemy producing a couple of recent papers. And this was a study doing immunoprecipitation and mass spectrometry. And it showed that there actually were 29 sites where tau was phosphorylated in brain homogenates. And there were 12 of these sites in CSF. And not everything in the brain was represented in CSF. As you can see, the C-terminal, that's very important in well represented in tangles, is not represented in CSF. Many of these sites in the mid-region are. When they quantified and looked at differences, there were several residues, p-tau 181, 217, and 331 in particular, and I've called these out, where there were changes. This is levels in the brain. This is non-AD CSF versus AD CSF for p-tau 181. Here's p-tau 217 and p-tau 231. So all of these might be more promising than some of the other phosphoepitopes. And so moving on to plasma p-tau 181, this was one of the exciting studies that came out earlier this year from the Swedish Biofinder study and a couple of other data sets. And taking you through what some of the data showed, first, there is a reasonably good correlation between plasma p-tau 181 using this particular assay system and CSF p-tau 181, but it was mostly in people who were amyloid positive. So if you were amyloid negative, there was not much of a relationship at all. And if one looked at the same sort of relationship with tau PET converted into Braque stage by different regions of interest, again, there was a relationship, but mainly in people who were amyloid positive. When this was applied to different categories, these are controls, people with MCI, amyloid positive and negative, and people who have MCI and AD and non-AD dementias, there's high sensitivity and specificity. Again, the levels in this particular assay, we're talking about 4 or 5 or maybe 7 or 8 picograms per mil, which is needs a very sensitive assay for measurement and detection against a background of about 2 picograms per mil. Nevertheless, these ROC curves are highly impressive. If one looks at when different forms of tau and other biomarkers change, this was a cross-sectional study from which some longitudinal modeling was done, comparing CSF and plasma from the same subjects. It turns out that A-beta 40 and A-beta 42 in CSF are among some of the earliest things to change. CSF phospho-tau and total tau started to change. They seem to change a little more dramatically after an A-beta PET threshold is reached. These are some other markers, neurogranin, YKL-40, neurofilament light. They seem to change somewhat later. In plasma, interestingly, p-tau tends to show a very similar profile as it does in CSF. Plasma A-beta 42 and 40 do show some declines, but the breadth, so the amount by which this differs from normal is much, much shallower than it is in CSF, where there's a much bigger effect size. So all of this suggests that we can use a combination of A-beta 42 and 40 in plasma, perhaps, but p-tau is possibly going to tell us something more. So I'd mentioned that there are different epitopes of tau, 217, 181, and 231. And this was a study looking at immunoprecipitation mass spec to identify p-tau 217 versus 181. And the major finding was that for 181, there was definitely an increase in people who were amyloid positive versus negative. But for 217, it looked as though there was less overlap. And this was the finding whether 181 was measured either through a mass spec assay or through an ELISA assay. when one looked at diagnostic value, even though this was a relatively small study, there was a higher ROC curve for P30 And so this has led to the ability to try and develop assays for 217 and 181 in plasma. And a recent paper based on the Swedish Biofinder study with a couple of other data sets suggested that levels of P217 were a little better than levels of P181 in distinguishing people with Alzheimer's disease and to some extent MCI versus controls. And these were the ROC curves. The ROC for P217 reached a very impressive 0.96, P181 reached 0.81. I would still say this is not quite definitive because there are other assays and antibodies that are still being developed for some of these different phosphoepitopes, and in particular, P231 may be similar to P217. Nevertheless, this looks like a very encouraging assay for plasma. If one turns now to plasma A beta, as I've shown from some of the other data, the problem is the dynamic range is a whole lot smaller. So it's possible to use the ELIXIS data assays to measure plasma A beta, and this was a study in which they were developed on the Roche platform, and there are significant effects. There is a reduction of A beta 42 in plasma, and there's not much of an effect of A beta 40. If you express it as a ratio of 42/40, it does a little better. If you also throw in information about other plasma biomarkers, in particular, if you throw in ApoE genotype and maybe add some information about plasma NFL, this used one of the older plasma tau assays, it's possible to come up with an ROC curve that's kind of in the 0.84 range. Obviously, this doesn't look quite as good as plasma p-tau did on its own, but if we're interested in detecting amyloid, this might give us some measurements. The other approach to measuring plasma A beta is to immunoprecipitate and capture A beta and then do mass spectrometry. This was a study from Randy Batesman's group, and the data has later been extended where there is a decrease in 42/40 in people who are amyloid positive. And this was a study from a Japanese group in which they had a very complicated immunoprecipitation procedure, and they showed something relatively similar. So it turns out that the immunoprecipitation mass spec assays for 42 and 40 at this stage seem to perform a little better than the ELISA and other assays, but lots of efforts are underway to try to produce better ones, and none of these perform quite as well as the CSF assays. So that's kind of a quick update on where we are in plasma. I wanted to cover trying to enlarge the biomarker portfolio and talk about some of our own interest in synaptic biomarkers in CSF. So this is another cartoon illustrating that there's a lot more than plaques and tangles, and we might think of synaptic biomarkers reflecting presynaptic and postsynaptic mechanisms. We might think of astrocytic biomarkers, markers reflecting blood-brain barrier function, microglia, axonal integrity, and these are all, most of these were developed by candidate biomarker approaches, deciding something was interesting and developing an assay. The other approach to identifying biomarkers is through omics to measure a host of different things and use unbiased approaches to figure out what matters and what doesn't. A number of these studies are underway, and I'm not going to talk about them today, but proteomics, metabolomics, things like microRNAs and exosomes are yielding some interesting findings. So why should we be interested in synaptic biomarkers in Alzheimer's disease? A lot of different reasons. Firstly, amyloid release depends on synaptic activity. One of the earliest findings, originally actually out of Bob Terry's lab and validated by all sorts of studies, including proteomic ones, is that the loss of synaptic markers in postmortem brain correlates strongly with cognitive measures. Various kinds of studies, MRI and EEG, have suggested that there is increased circuit excitability in MCI and preclinical AD. And then there are pathogenic mechanisms. So oligomers of A beta, aggregates of tau, a whole bunch of other neurodegenerative processes may all result in synaptic toxicity. And then if that wasn't bad enough, activation of astrocytes and microglia may damage synapses, for example, through complement release or synaptic pruning. What should we choose to measure? It's actually not an easy choice. There are a bewildering number of synaptic proteins that have been identified as shown in this cartoon. And what I'm going to show you comes out of some candidate biomarkers identified and selected for various reasons. We've focused on being able to measure a postsynaptic marker, neurogranin, presynaptic SNAP25, and neuronal pentraxin 2, which is secreted. So a little bit about neurogranin. It's a dendritic calcium-related protein. It's increased in CSF in AD. This was a study from 2015 showing some increase that might even begin at the MCI stage, a fair amount of overlap as well, a hint of a change in people who are normal and are at risk for Alzheimer's disease. Of interest, neurogranin correlates strongly with tau and p-tau in CSF regardless of diagnosis, and perhaps the processes by which neurogranin is released into CSF overlap with processes by which tau is released. Neuronal pentraxins are interesting proteins. We've been collaborating initially with Paul Worley to study them further. There's a family, and NPTX1 and NPTX2 are secreted presynaptically. There's a receptor, NPR, neuronal pentraxin receptor, which is tethered and then cleaved. And these pentraxins help to cluster AMPA receptors, which helps to regulate how excitable cortical circuits might become. And they have roles in general synaptic maturation. What got us interested were some data that Paul Worley originally demonstrated showing that NPTX2 and NPTX1 were markedly decreased in brain homogenates from different regions in people with Alzheimer's. There was a hint of a finding in people who were asymptomatic but had some AD pathology. So we collaborated and found some impressive findings. This is Western blot analysis of CSF where there's a marked reduction of NPTX2 in Alzheimer's disease, some change in NPTX1, and not much change in NPTX receptor. And so an ELISA was developed, and we've carried out a number of further studies showing that CSF NPTX2 is decreased in Alzheimer's disease, and it relates to cognition. And we've been interested in whether it may help to predict progression in MCI or mild Alzheimer's disease. So these were studies that Dennis Smirnov helped us with enormously, in which we've looked at NPTX2 in the UCSD ADRC. This looks at progression on a number of different cognitive markers, and also on the CDR sum of boxes in our cohort. And these are models in which we've thrown in lots of other things. CSF A beta 42 and tau are also in these models, and we still find that adding NPTX2, those people who have low NPTX2 have faster progression on all of these different measures. And when we looked at ADNI and threw in a bunch of different CSF biomarkers, NPTX2 had value in predicting progression from MCI to Alzheimer's disease, even when we included A beta 42 and tau, and when we included NFL, that actually didn't have any extra prediction. So NPTX2 has now been measured in a number of different disorders, and of interest, it's also decreased in Down syndrome and frontotemporal dementia. So while it might not be specific, it might be telling us something about synaptic function. So in the last few minutes, I'm going to talk about yet another aspect of biomarkers, namely protein seeds. What I've talked about so far is simply big lesions like plaques and tangles, but a common theme across neurodegenerative disorders is protein misfolding. And it is believed that during preclinical stages of a host of different disorders, there is a buildup of misfolded proteins. Although not everybody believes this, there is evidence to suggest that misfolded protein seeds may spread from neuron to neuron, and this has been called prion-like spread, whether the seeds themselves cause the misfolding or whether they trigger some sorts of events that misfold the the candidate protein is still under some debate. Nevertheless, as a result of these misfolding events, there's neurodegeneration and pathological responses. And then depending on which protein has been involved, which cell types are involved, and which brain regions are involved, there will be different symptoms. So Alzheimer's is associated with misfolding of amyloid beta protein and tau, Parkinson's with alpha-synuclein. ALS is associated with a number of different kinds of protein seeds. TDP43 seems common to all forms of ALS. In some forms, the protein FUS is associated with misfolding, and in some SOD1 is the culprit. And then in Huntington's disease, Huntington protein misfolds. So a question with regards to biomarkers is, instead of looking for indirect evidence of neurodegeneration, is it possible that we can actually identify the protein seed that may be the culprit of neurodegeneration against a background of a whole lot of normal proteins, as I hope I'm trying to illustrate in this cartoon? It's very difficult to detect seeds. They are not, or they haven't been detectable by routine assays such as ELISAs and so forth. A technology that has initially started in the world of prions and Creutzfeldt-Jakob is RT-QuIC, or real-time QuIC, which is a little bit like a protein equivalent of a PCR reaction. One adds fuel to a protein, shakes or agitates the protein, and over time the protein might form seeds that are detectable. This has been applied by several laboratories to be able to detect alpha-synuclein as pathological seeds in a microplate. And so this is a cartoon showing that this can be done on a 96-well plate. The idea is to add some of the fuel or monomeric alpha-synuclein, add an amyloid-sensitive dye, thioflavin T is usually used, a reaction mix of a couple of other ingredients, and then the test sample, which might be brain homogenate as a positive control, or a CSF sample. And then this is subject to many cycles of agitation and then resting. And the idea is that over time, if there are seeds in the test sample, they glom onto the monomeric synuclein in this case. They recruit it and convert it into further seeds. It fragments and elongates and sets off a chain reaction. And over time, this reaches a point where it takes off a lot and includes thioflavin, and this can be read out as a positive. So there's a lot of exciting data in the Parkinson's world showing that RT-QuIC, or some variants, it's also been called PMCA, can yield similar data across laboratories. And what this slide shows are data from 3 of the laboratories that have contributed, Claudio Soto, Alison Green, and Byron Coy. And these were a series of samples from the BioFind study, 105 patients with at least moderate to severe Parkinson's disease and 79 controls. And the results were more or less concordant across all 3 labs. Some of the controls actually showed positive reactions, and of interest, at least one of these controls has later gone on to develop Parkinson's disease. A small number of Parkinson patients have shown negative reactions. Overall, there's a sensitivity, it's somewhere at least around 95%, which is very encouraging, and specificity somewhere in the 80 to 90% range. The largest study using RT-QuIC for alpha-synuclein that's been carried out to date was by an Italian group that measured over 400 CSF samples. And this illustrates a summary of the findings. They found that of interest, there are positive reactions in a small number of Alzheimer patients. These are clinically diagnosed, and as many of us know, some of these patients may harbor comorbid synuclein pathology. And we're in the course of pursuing this sort of question using CSF samples from the ADRC. But in general, AD patients do not show positive RT-QuIC for synuclein. PSP is negative. Multisystem atrophy using this particular assay was negative. There are some other assays where MSA gives a weak positive. And here are a host of different Parkinsonian disorders, including very early stages. This is idiopathic REM sleep behavior disorder, where every patient showed a positive reaction. The extent of positivity doesn't seem to correlate with different stages, and we're not sure that this is a quantitative rather than an on-off kind of assay. Here were data on sensitivity and specificity across different categories. So the bottom line was that there was very high sensitivity and specificity in this particular study, which is very encouraging for synuclein disorders. So far, there's some work on trying to do RT-QuIC for alpha-synuclein in plasma, with a hint that there are some positive findings, but that the assay needs to be refined. It also is possible to try to apply RT-QuIC for tau, and Byron Corey's lab has done so. These are some examples of looking at a 4-repeat tau assay for RT-QuIC done in a number of different post-mortem brain homogenates, and in particular, some disorders where there is selective accumulation of 4-repeat forms of tau, PSP, for example, corticobasal syndrome, and some tau mutations are associated with very strong reactions. Some other disorders, Alzheimer's disease, do have some weak reaction. In Alzheimer's, there is a combination of 4-repeat and 3-repeat tau in the tangles. So while this is very promising in post-mortem brain, we're not going to wait around for people to die to be able to hope that that's the only time we can confirm a diagnosis. There have been attempts to try to develop this in CSF, and the initial assays showed about a 50% positivity rate for the 4R tauopathies, which is not good enough. So further efforts are underway to try to increase the sensitivity or do something a little different. But so far for tau, we are not at anywhere near the same level of detection of seeds as we would be for alpha-synuclein. So in summary, I've covered quite a lot of ground. And what I'd like to leave you with is that the core CSF biomarkers, A-beta 42, or the ratio of 42/40, tau and phospho-tau, have diagnostic value for Alzheimer's disease, which is independent of stages of clinical stages of disease, and that these map onto the A and N parts of the ATN framework. Neurodegeneration, even though it's non-specific, occurs in other disorders besides Alzheimer's disease, is definitely worth digging into because one can identify markers that correlate with cognition and maybe aid in prognosis. I'm going to strongly encourage deeper biomarker discovery to identify a broad panel of markers for neurodegeneration and to add markers for inflammation, vascular changes, synuclein, and so on to increase our armamentarium to diagnose Alzheimer's and related disorders. And then very excitingly, plasma and serum biomarkers are finally reliably detectable. And not only that, they have value at the very least as screening tests for Alzheimer's disease, maybe even a little better than screening tests, and as acute damage markers that are relevant to conditions such as traumatic brain injury. And then biomarkers for pathogenic seeds, which I think are relevant for therapeutics, are detectable at this stage in CSF for alpha-synuclein. So I'd like to end by thanking a large number of people whom I've collaborated with in these and other studies. And I'd also like to thank a number of sources, NIA, VA, ADDF, Michael J. Fox, and the Alzheimer's Association, who have funded a lot of this work. Thank you very much, and I would be happy to take some questions.
Well, thanks very much, Doug. I'll jump in real quick if you don't mind. Just wanted to see, do you think the IP mass spec is scalable, or do we need to have assays that are developed?
IP, I don't believe, so when you say scalable, it's scalable by custom laboratories that have enough mass spec machines and so forth. And I believe that that would be an approach that could be done reasonably. Having said that, IP mass spec is much more complex than some of the classic sandwich type assays. And I think we're highly interested in seeing whether there are additional ultrasensitive assays using antibodies that might improve on some of the Quanterix or Simoa methods, or whether there are different antibodies that are better than the existing ones, and see where that leads us.
Hey, Doug, Stuart here. Great talk. Wanted to ask you if you've seen the new Dominic Walsh NT1 tau data that was published in the Annals of Neurology this week.
So I was aware of that data. It looks like a replication of the first study that we did with Dominic. And it's interesting data. Haven't seen a head-to-head
that only certain epitopes of NT1 tau, and therefore only certain antibodies work. And that's why many people had failures. And it brings up the problem that we probably have to use very specific antibodies. And if they're polyclonals, they get exhausted. So it really calls into question some of the other work and how.
Right. So actually, most of the tau assays have been developed using monoclonals. But the big question for detecting total tau or tau fragments in plasma is that there are lots of different fragments, and many of them are likely degraded by proteases. And it's possible that Dominic Walsh's antibody detects some of these fragments, or some core piece of fragments that still remain even after the degradation. So it's an exciting finding and a promising reagent, and we need to put the findings of phospho-tau into perspective, which hasn't yet been done.
Hi, Dr. Galasko. I was curious about synaptic dysfunction. Usually happens before cognitive decline. So have you seen any of the synaptic markers prior to cognitive impairment?
So that's a great question. So far, I'm not aware of any marker being consistently altered in CSF before the advent of cognitive impairment. There are a few hints for some of the synaptic markers. We've measured some other synaptic markers. In some people's hands, if you have longitudinal data on phospho-tau, for example, it might begin to change. And I think that it's a difficult area and a difficult question. I think that we are either going to need an ultrasensitive kind of assay or a panel of markers to be able to detect what I think are the very, very small changes that occur in these early stages. But there's certainly a lot of interest and work ongoing.
I'm going to read off a question here in the chat. It says, would phenotyping patients based on biomarkers improve on the historical clinical diagnosis framework?
I believe it would. So I think we've all seen diagnostic errors when we make a clinical diagnosis of Alzheimer's disease, even if we base it on our very best clinical characterization, especially for atypical forms of Alzheimer's, for example, progressive aphasia, or younger onset Alzheimer's, what we think is Alzheimer's, we can be wrong. For late life Alzheimer's, we've had mistakes where, for example, hippocampal sclerosis has been the primary and major pathology underlying what looked like an amnestic presentation. So I think biomarkers can, if we even narrow it down to A and T, biomarkers can improve on clinical diagnoses, even in those few examples.
I had a quick question, Doug. That was such a great talk. Thank you. With the blood-based biomarkers, the new tau markers, we see sex differences in CSF tau, we see sex differences in PET tau. I haven't seen anything yet specifically with the blood data. Do you have any awareness if anyone's looked at that in a sex stratified way?
So I think these are early days, but what I have seen just from looking at the data from Swedish Biofinder is that they did not have gender-associated differences. I don't know how large these differences might or might not be, or whether they might only emerge if you looked at the stage of Alzheimer's disease rather than MCI, for example. And I think as as we see more and more data, people will begin to peer into this. In our small study looking at the Dominic Walsh antibody with the NT1, we didn't see a change that was gender related or sex related, but that was a fairly small study.
Yeah, we in CSF and PET, we tend to see it at the MCI stage specifically. Thank you.
I also want to say really excellent talk, Doug. Since you know a lot more about the background stuff that's not necessarily published, my impression is that when they're talking about healthy controls, it's really healthy controls. Do you know if anybody goes back and checks the results against a more normal population that we might typically see on the street with some head injuries, maybe a little drug abuse, alcoholism, sort of normal people?
So those are great questions, and that research needs to be done to really validate some of these markers. The one I would be concerned about, in particular for plasma, is neurofilament light, where all sorts of things can bump it up a little bit. We don't know, for example, with some of these markers, whether some subtle alterations in renal function may alter clearance and bump up levels, or changes in liver function could make a difference. I think for plasma A beta, people have looked at certain things like lipid levels that don't seem to make a difference, but a lot more work and careful work will need to be done to decide that that cutoffs are legitimate and that we can use these without a number of other factors in interpretation.
Yeah, great talk with Paul here. I just was thinking if endosomal proteins could be, kind of a biomarker, because early endosome pathology is observed well before A beta and tau pathology are kind of visible.
So that's a great question. And the question for any biomarker that we find, I'll start with CSF, is how does it get into CSF in the first place? So a lot of things could be released, for example, non-specifically if a cell dies. And I think to some extent, certainly in medium to later stages of Alzheimer's disease, some of what we're measuring might reflect cell death. And that could release all sorts of things. For other biomarkers, some may be secreted through very well-defined processes. So for example, A beta is secreted as the result of a number of well-known enzymatic cleavages. How tau gets into CSF is not very well understood. It's believed to be an active process, and there are a number of ongoing studies about non-classical secretion of proteins and what some of the pathways and mechanisms could be. And they're very interesting. How might endosomes get out of cells and into CSF? Well, it's not clear, but there may be some mechanisms where cells under stress might be shedding them. And then how would we detect the proteins? So there's been a little bit of work on lysosomal proteins in CSF, and some of them are measurable, but so far, from what I've seen, there are not big changes in Alzheimer's disease versus controls. So I think that these are early days, and I think looking into proteomic data sets and trying to come up with very specific targeted assays would be my suggestion to try to identify whether your own favorite protein or pathway is represented.
There's another question in the chat here. Do you think biomarker blood screening will eventually be used in practice despite the dilution effect?
So I think the answer is yes. And I think many of us are wondering about how a blood test might be used. So would it be used before any kind of history or exam or cognitive testing, which I think many of us would be opposed to, because it would make it much more difficult to interpret both a positive and negative result? Or would it be used to guide who gets referred upstream to someone with some expertise for a more detailed kind of investigation? I think studies are going to have to look at this and come up with diagnostic pathways and prove that certain things have value as a result. Certainly plasma biomarkers have the potential to save on having to do PET imaging and potentially save on having to do lumbar punctures.
There's one more in the chat here. It says, have you seen any biomarkers changes from INS from AD fibroblasts?
Okay, so ions are induced neurons from AD fibroblasts. That's an interesting question. We've been involved in studies using fibroblasts from Alzheimer's, making them directly into neurons to try to figure out if aging makes a difference. We haven't been looking for biomarkers per se, but rather looking at whether there are changes in the cells or pathways interrogated by things like microRNA, at least RNA-seq analysis and things like that. Having said that, there are some changes that suggest that maybe they could be reflected by biomarkers, but the numbers of INs in these studies are pretty small, and we'd have to take these as insights to then go back and look at things like CSF and plasma. Okay, so I think it looks like we're also out of time. So thanks everybody, and I'd be happy if people do have additional questions and suggestions, please contact me.